A microarray is a laboratory tool in which a pattern of single-stranded DNA probes are immobilized on a surface called a chip or a slide. Microarray assays large amounts of biological material using hybridizations to detect a specific DNA or RNA in a sample. Microarray technology evolved from shorten blotting. The concept behind microarray was first introduced by Ling and his colleagues in 1984, where they analyzed retroviral long-terminal repeat in a mouse colon tumor. The principle behind microarray is the hybridizations between two DNA strands by forming hydrogen bonds. Fluorescent label target sequences that bind to a probe sequence generate a signal that depends on the strength of the hybridizations determined by the number of paired bases. There are many types of microarray that have been used in the study. For example, DNA microarray, protein microarray, peptide microarray, tissue microarray, and antibody microarray.
DNA microarray procedure consists of 6 steps. Step 1 is to collect sample which can be from a variety of organisms. Step 2 is RNA isolation, where RNA is extracted from the samples. mRNA is then isolated from the rRNA and tRNA. Step 3 is to create a labeled cDNA. A labeling mix is added to the RNA. The primer and the reverse transcriptase bind to the mRNA first, then add the fluorescently dyed nucleotide, creating a cDNA. Step 4 is hybridization. The cDNA created is applied to the microarray plate. When comparing two samples, apply both samples to the same plate. The single-stranded DNA will bind to the cDNA present on the plate. Step 5 is detection of relative intensities of fluorescence under microarray scanner. The laser causes the hybrid bonds to fluoresce, the camera records the image produced, and the computer allows us to view the results and store our data. Step 6 would be the analysis of data. This is the example of analysis of data. Red fluorescence shows the upregulation of gene, and green fluorescence shows the downregulation of gene.
What are the applications of microarray? Microarray can be used as a gene expression profiling tool. Gene expression tool. Microarray can be used to study when, where, and to what magnitude genes of interest are expressed. It can be used to measure changes in the multi-gene patterns of expression to better understand about regulatory mechanisms and broader bioactivity functions of gene. Microarray also can be used for disease diagnosis. Researchers are able to classify the types of cancer on the basis of the patterns of gene activity in the tumor cells. For drug discovery, comparative analysis of the genes from a diseased and a normal cell will help the identification of the biochemical constitution of the proteins synthesized by the diseased genes. Microarray also can be used as a comparative genomic tool. It can be used to analyze genomic alterations such as sequence and single nucleotide polymorphisms. In microarray gene mutation analysis, it can be directed to characterization of genetic differences among microbial isolates, especially the closely related species.